this chapter, we will start to look at meiosis in the sexual life cycle. So up until now, what we looked at was mitosis, which was how the non-sex cells divide. So this is going to look at how the sperm and the egg divide. Once we start looking at meiosis, where we're combining the information from two individuals to create a new life form, it's going to automatically bring in heredity and genetics. Heredity is the transmission of traits from one generation to the next. Genetics is the study of heredity and hereditary variation. So the offspring are going to acquire their genes from their parents, and they're going to do this because they inherit the chromosomes. The genes are located on the chromosomes. Genes are the units of heredity. They're going to program for specific traits. Your gametes are the reproductive cells in the plants and animals, the sperm and the egg. They'll transmit genes from one generation to the next. Your somatic cells are all the cells in the body except the gametes. In humans, these are going to have 46 chromosomes. And the locus refers to the specific location of the gene along the chromosome. So when we compare sexual and asexual reproduction reproduces offspring asexually, and their offspring are going to be an exact genetic copy of the parent. Just a single individual is the sole parent. So they will produce clones, which are groups of genetically identical individuals. With sexual reproduction, you have two parents that are going to give rise to an offspring. That offspring is going to be a unique combination of the genes inherited from the two parents. So when we look at the life cycle, it's the time from generation to generation. The sequence of stages in the reproductive history of an organism from conception to making their own offspring. So we can look at the chromosomes in an individual. A karyotype is just an image of the chromosome. They get arranged in pairs, starting with the longest chromosome to the shortest, and then finally with the sex chromosomes, and they're in an ordered display. So you try to pair them up where they're going to have similar staining patterns and similar centromere locations. So homologous chromosomes are going to be two chromosomes composing a pair with the same length, same centromere position, and same staining pattern. So here you've got the two homologous chromosomes. For your sex chromosomes, these are going to determine the individual's sex. XX is female, XY is male. Only a small part of that chromosome is homologous because there is a significant size difference between the X and Y chromosome. Your autosomes are the non-sex chromosomes, so it's everything else. Your diploid cells are going to be any cell that has two chromosome sets, and we abbreviate this as 2N. So for humans, the diploid number is 46. Your haploid cells are going to be the sperm and egg, or your gametes. They're going to have just a single set of chromosomes. So for humans, the haploid number is 23. So the behavior of the chromosome sets in the human life cycle is going to determine how you get a new individual with a combination of genetic material from the parents. So during fertilization, you have the union of two gametes. It's going to result in fusion of the nuclei. The zygote is that fertilized egg. In order to be able to do this, you need to undergo meiosis to reduce the number of sets of chromosomes so that that zygote will have just one complete set that a normal adult does. So there is some variety in the sexual life cycles. It is a little bit different if you're looking at plants, animals, or the fungi and the protists. So this is easier to look at this diagram. Here we've got the animals. So what we're going to do is start here with meiosis. In an animal, it undergoes meiosis to make its gametes, the sperm or the egg. These are going to be haploid cells. These remain single-celled. You'll never see a multicellular sperm in an animal or a multicellular egg. These will fuse to undergo fertilization and produce the zygote. The zygote is going to undergo mitosis to make the diploid multicellular organism. This is the dominant stage of their life. Over at the other end of the spectrum here, where we've got the fungi and the protists, they will undergo meiosis to make gametes here. These will undergo mitosis themselves even though they are haploid individuals, they will undergo multiple divisions to become a multicellular organism, or in the case of a unicellular organism, it will be the dominant part of their life cycle. Then they can undergo fertilization, having two of them fuse together, and then will form the diploid zygote that will immediately undergo meiosis again. With plants, they kind of take a little bit of both worlds. So a plant can undergo meiosis to produce two haploid cells. Those haploid cells in this gametophyte generation can undergo mitoses to become multicellular organisms. Then those gametes can undergo fertilization and fuse together to make a zygote. That zygote can also undergo mitoses to become a diploid multicellular organism in the sporophyte generation. So with the plants, you have two different times of being multicellular organisms. So with these, you can actually have two different plants look significantly different, even though they are the same species, whether it's the gametophyte generation or the sporophyte generation. So the goal of mitosis was to divide the nucleus. The goal of meiosis is to reduce the number of chromosome sets from diploid to haploid. So we have two main phases, meiosis 1 and meiosis 2, that will do two divisions. This will result in 4 daughter cells that only have half the genetic information, half the chromosomes as the parent cell. So when we have sister chromatid cohesion, this is the association of two copies of one chromosome along their lengths. You have one duplicated chromosome. With homologous chromosomes, you have individual chromosomes that were inherited from different parents. They're going to have different alleles. The allele is the version of the gene. So the sister chromatids are exact copies of each other. The homologous chromosomes are the same chromosome, but they were inherited from different parents. So when we look at interphase, you're going to have your pairs of homologous chromosomes in the diploid parent cell. They're going to duplicate this pair of homologous chromosomes, and then you will have the sister chromatids paired together and the diploid cell with the duplicate chromosomes. During meiosis 1, the homologous chromosomes are going to separate. Here you'll have haploid cells with duplicated chromosomes. During meiosis 2, the sister chromatids are going to separate. Here you'll have haploid cells with unduplicated chromosomes. So when we compare what happens in meiosis and mitosis, if we look at the DNA replication, in both of these, this is going to occur during interphase before meiosis or mitosis begins. Mitosis will undergo one division, and it will include prophase, metaphase, anaphase, and telophase. In meiosis, you undergo two divisions. Each division will have a prophase, metaphase, anaphase, and telophase. They would be known as prophase 1, metaphase 1, anaphase 1, and telophase 1, and then prophase 2, metaphase 2, anaphase 2, and telophase 2. When we look at synapsis of the homologous chromosomes, this does not occur in mitosis because there are no homologous chromosomes. However, this occurs during prophase 1, along with crossing over between the non-sister chromatids. The resulting chiasmata here is going to hold the pairs together due to sister chromatid cohesion. When we look at the number of daughter cells in the genetic composition, in mitosis you get two daughter cells. They're each diploid, and they're each going to be genetically identical to the parent. In meiosis, you're going to get 4 daughter cells. Each of them will be haploid with half of the chromosomes as the parent, and they will be genetically different from the parent cell and each other. So the role in the animal body, mitosis is going to allow them to become a multicellular adult. In meiosis, this is only used to produce the gametes. It reduces the number of chromosomes and introduces the genetic variability into the gametes. So there's 3 events that are unique to meiosis 1, synapsis and crossing over, where you duplicate the homolog pairs and form the synaptonemal complex during crossing over. Homolog pairs are only going to occur during meiosis. The homologous pairs line up at the metaphase plate. This is going to be only in meiosis because you only have homologs in meiosis. So rather than having the individuals line up, you would have the homologs line up. And then the separation of the homologs, your duplicated chromosomes of the homologous pairs separate, but the sister chromatids are going to stay together. They're able to do this because the enzyme that cleaves the cohesin is actually released in two parts in anaphase 1 and 2. The chiasmata is going to hold the homologs together as the spindle forms during the first mitotic division in the onset of anaphase 1. The cohesin is released, and this allows the homologs to separate. So meiosis 1 and meiosis 2 are also known as a reductional and equatorial division. So the reductional division is meiosis 1. It halves the number of chromosome sets per cell. The cell goes from diploid to haploid, splits the homologs, and now you just have the sister chromatids. During meiosis 2, you have the equatorial division. Here the sister chromatids separate and produce the haploid daughter cells. So one of the important things about sexual reproduction is the increase in genetic variation among the offspring. The behavior of the chromosomes during meiosis and fertilization are going to give most of the variation. So we have the independent assortment of chromosomes. So during metaphase 1, the homolog pairs are going to consist of one maternal and one paternal chromosome. So each may pair with either a maternal or paternal close to a given pole. You've got a 50-50 chance that each particular daughter cell will get the maternal or the paternal chromosome. So here you've got a 50-50 chance of getting the maternal or the paternal. Again, at this one you have a 50-50 chance of getting the maternal or the paternal. So this is going to occur independently for every pair. So the possible number of combinations in humans, you've got 2 possibilities for each pair, and you have 23 haploid pairs. So that's 8.4 million possible combinations of the chromosomes. So if you were to look at your sperm or your eggs and you wanted to know if you could produce an exact duplicate, it means there's a 1 in 8.4 million chance of producing an exact duplicate based on just the chromosomes alone. So here you can see if they split this way, you're going to get these 4 combinations. If you have this second possibility where you have one from mom and one from dad, then you're going to get these possible combinations. So in addition, we can have crossing over that will produce recombinant chromosomes. So here the individual chromosome is going to carry genes from 2 different parents. An average of 1 to 3 crossing over events occurs per chromosome pair, and this begins early in prophase 1 as the homologous pairs of chromosomes pair with the genes precisely lined up. So in the crossover, the DNA of non-sister chromatids, it's broken by specific proteins at precisely corresponding points so that they'll have a crossover in the exact new combination of maternal and paternal alleles. In metaphase 2, the recombinant chromatids can be oriented in 2 alternate ways to further increase the genetic types of the daughters. So here you can see where you've got the chiasma joining those 2 together, and you have a little piece of each of the chromosomes that is swapped places. So this is crossing over, and the result is 2 new recombinant chromosomes. So here when you look at the daughter cells with crossing over, now each daughter cell is unique. In addition, we have random fertilization. So in humans, it's going to take a male and a female gamete in order to produce your zygote. So each of those represents 1 in 8.4 million possible combinations. So when you fuse the zygote, you're going to have 2 to the 23rd times 2 to the 23rd, which would be 70 trillion possible combinations. So even in the event that you were able to produce 2 gametes with the same exact set of chromosomes, the odds of the second person doing that as well, to have 2 zygotes that are exactly the same, are extremely unlikely. So the evolutionary significance of the genetic variation, this is going to put more possible combinations into the gene pool.